Lesson 25, Science 7, and today is the day to see if those two girls are related. Grab your pencil, your journal slip, your science journal, and later on you're going to create some paper chromosomes using a red and blue colored pencil for color coding. So let's find out if those two girls are related. Here, listen.
These pretty teens are best friends, and they're also something else. Something that may surprise you.
We're twins.
Believe it or not, they're twins.
Yep, we're twins.
We're
Same
twin
mum,
sisters.
same dad.
You heard right. They have the same mom and dad. What are the odds that one would be black and one white?
A lot of people are just in shock.
Lucy and Maria Aylmer live in Gloucester, England. Maria has a caramel complexion, brown eyes, and thick curly black hair. Lucy has pale white skin, ginger red hair, blue eyes, and freckles. Most people have the same reaction when they hear they are twins.
They say, how did it happen? We don't believe you.
So how did this happen? Better yet, maybe you can relate in some way. It might be that you look nothing like your sibling or exactly like your sibling. Or maybe some say you're a spitting image of your mom, look just like your dad, or closely resemble a grandparent, aunt, or uncle. Or maybe you feel like you look very different from everyone in your family. These phenomena spark many questions. Here are pictures of my two children, Giovanna and Dominic, on the day they were born. We joke in our family that they looked so alike at birth. Their mouths, their chin, their complexion, and here's a picture of them older. We still say that you can tell that they're brother and sister. Stop and think about your own family. Who do you look like the most? The least? An obvious question you might have is how can siblings be so alike and others so different? How does that happen? To figure this out, we need to know how living things inherit traits. Believe it or not, there was a man by the name of Gregor Mendel who was interested in this very question. Mendel was an Austrian monk who lived in the mid-1800s and wanted to figure out how pea plants inherited certain traits. Why pea plants, you ask? Well, while living at the monastery, he spent most of his time in the garden, and he noticed that these plants weren't all the same. They had different traits. Most notably, he saw that some plants grew really tall, while others grew pretty short. Mendel wondered how plants inherited this trait of height from their parents. So to find out, he conducted experiments over many years. And by mating plants and observing how the offspring or the baby plants came out, he was able to answer this question. Today, you're going to get to simulate Mendel's experiments. But before we get started, you need to know that each organism has a unique number of chromosomes. Humans, like me and you, have the same 46 chromosomes in each and every one of our cells. 23 of these chromosomes come from our mother, and the other 23 from our father. That's why you see them on the screen arranged in pairs. Pea plants, on the other hand, have 14 chromosomes in every single one of their cells. 7 chromosomes are inherited from the mother plant and 7 chromosomes from the father plant. For each plant, we're going to use paper strips, like you see here, to represent the chromosomes. Remember, these chromosomes are nothing more than coiled up DNA. And if we uncoil the DNA enough, we can read the strings of A, C, T, and Gs. These sequences of nitrogen bases form a gene, and this gene can make proteins that can contribute to the plant's height. For the purposes of today's activity, let's say that the gene for height is on this first pair of chromosomes right here. Let's create this chromosome pair for the mother first. To do this, you can either cut out strips on the last page of your journal slip or use a piece of blank computer paper to make your own, like you see on the screen. Now take a red colored pencil and write or outline the uppercase T on each chromosome. Uppercase T is the shorthand for the tall gene. So mom has 2 tall genes. We call her a purebred. Now let's create Dad's chromosome pair. Cut out the strips or make your own. This time, take a blue colored pencil and write or outline lowercase t on each chromosome. Lowercase t is the shorthand that geneticists use for the short gene. So Dad has 2 short genes. He's also considered a purebred because he has 2 of the same genes. You're all set to do experiment 1. When Mendel did this experiment, he called these plants his P generation. And remember, these were purebreds, meaning that each plant had 2 of the same genes. You're now going to make these 2 parent plants by randomly choosing 1 chromosome from Mom and 1 chromosome from Dad to pass on to the baby plant. It's random which chromosome is being passed on, so no peeking. You should have your chromosomes turned over like I do. Now, match them together and record the gene combination of the baby plant by putting a tally mark in the correct location of your data table. But this is only the first offspring. I want you to mate Mom and Dad 9 more times so that they have a total of 10 offspring. This means that you should give each parent back their chromosomes, give it a little shuffle, and randomly choose again. Go ahead and carefully complete experiment 1 now. I'll wait while you mate these plants. Okay, let's see what you figured out. Mendel called these offspring his F1 generation. What did all these baby plants look like? Yep, according to your tally chart, you can see that these offspring look tall. And the gene combination was always uppercase T, lowercase t. So Mendel would have considered these offspring hybrid plants because the baby plant has 2 different genes, 1 tall gene and 1 short gene. Now, Mendel was thoroughly confused. None of the offspring came out short, even though one of the parents, the father, was short. What happened to that trait? Had it disappeared? All right, so he decided to do another experiment, experiment 2. This time he took 2 baby plants from experiment 1 and mated those. Therefore, you need to make 1 parent plant a hybrid, like you see on your screen, and the other parent must also be a hybrid. To do this, cut out the paper chromosomes for experiment 2 or make your own using white computer paper. Then use your colored pencils to write or outline Mom and Dad's genes. Are you all set? Now, turn the parent chromosomes over so that you can't see the genes being passed on. This is important because inheritance is random. Parents don't get to choose which genes are passed to their children. Now randomly choose 1 chromosome from each parent. This is baby number 1. Look at the gene combination for this baby plant and record it in your data table. Okay, reset the Mom and Dad chromosomes and turn them over. You should even make sure to give each set of chromosomes a shuffle. You shouldn't ever know where the genes are. I want you to have these parents make 99 more offspring for a total of 100 baby plants. Yes, you heard me, 100 baby plants. Don't go over or under, 100 offspring exactly. And no peeking when choosing chromosomes. Pause this screencast and do experiment 2 slowly and carefully. Remember to accurately tally your data as you go along. If you want to have someone else in your household help you mate or tally, you can do that. Just work carefully and slowly. I have all the time in the world. Believe me, I'm not going anywhere. I'll be here when you get back. So again, I'll wait while you mate these parent plants. Finally, you're done. Good work. Let's analyze these results. This is where the magic happens. First, let's calculate the ratio of tall to short plants. Remember, tall offspring are plants that are uppercase T, or uppercase T, lowercase t. And the only way to be short is to have 2 lowercase t's. or 2 short genes. There are also some directions on your journal slip to help you. Now, I want you to calculate this ratio as a percent. This should be easy since the numbers are out of 100. So if 50 of your plants were tall, then that's 50%. Now here's the best part. I bet I know which of these 4 ratios best matches your results. First, you choose. Okay, are you ready? Really, I bet I know. I know you're ready for the magic. I bet your ratio is closest to 75% tall and 25% short. Now, I bet you're wondering how I knew this. Well, the answer's simple. It has to do with the rules of probability, chance. Mendel figured out that there are only so many ways that the genes from mom can combine with the genes from dad. The possibilities get shown in a special chart that you see on your screen called a Punnett square. Here, look. I set up mom's chromosomes along the top. She has 2 of them. And dad's chromosomes right here. Now, it could be that when these parents go to have an offspring, mom gives this chromosome with her tall gene, and dad gives this chromosome with his tall gene. And the offspring would come out tall. That's the first possibility. But another possibility is that mom gives the chromosome with her short gene, and dad gives the chromosome with his tall gene. In this case, the offspring would still come out tall. That's possibility 2. Or it could be that mom passes on the chromosome with her tall gene, and dad passes on the one with his short gene. And again, in this case, the offspring would look tall. That's possibility 3. Or both parents might pass on the chromosome with their short gene. And this is possibility number 4. There are 4 possible outcomes every time these 2 parents mate and have a baby plant. Since there are only 3 ways out of 4 to have a tall offspring, then you can expect an offspring to be tall 75% of the time. And since there is only 1 way out of 4, right here, to have a short offspring, you can expect an offspring to be short only 25% of the time. But remember, this is all chance. I could have done this experiment and observed 100 baby plants that were all short. While it's theoretically possible, in actuality, that is extremely unlikely, given the rules of chance. Now, no matter how many times Mendel did experiment 2, he always observed the same percent ratio, 75% tall and 25% short. Mendel analyzed thousands of offspring, not hundreds. He figured out that certain genes are dominant, meaning that they always show up when inherited, like the tall gene. And others are recessive, means they usually get hidden when the dominant one is present. This is why shortness seemed to disappear in experiment 1. Since all those baby plants were hybrids, the short gene was hidden by the dominant one. Now, congratulations, you have finished your first investigation on how traits are passed on. You're probably seeing that the parents are passing on chromosomes, and those chromosomes have our genes. For today, please submit the journal slip Mendel's Garden and use that information from the investigation to take a Google form. The link for the Google form is posted on Schoology. Until tomorrow, keep thinking and keep learning.